Dear students, now we are going to discuss where does photosynthesis take place? What are the areas, are the structures, cell organelles, where are in which photosynthesis occurs? So simply, chloroplast. As we are studying photosynthesis in higher plants, definitely we have to know the structure of a cell organelle which is concerned with photosynthesis. If you take algae, bryophyta, pteridophyta, gymnosperms, and angiosperms, all these are eukaryotes. Maximum number of photoautotrophs. In all these, the cell organelle concerned with photosynthesis is chloroplast. In the case of blue-green algal members, cyanobacteria, chromatophores are present. Bacteria also, chlorobium, chromatium also with chromatophores. Go back to the unit of life. In that case, under the heading prokaryotes, under the heading nutrition towards right side, first, second line or third line, chromatophores are present in blue-green algal members, are cyanophysi members. They are prokaryotes able to perform oxygenic photosynthesis. Generally, oxygenic photosynthesis is present in photoautotrophic eukaryotes. But there are certain number of prokaryotes with oxygenic photosynthesis. For that, the best example is blue-green algal members, cyanophysi members. If they question you, what are the prokaryotes with oxygenic photosynthesis? Go for blue-green algae, cyanophysi. According to Whittaker, they are no more algal members. They are monerans, prokaryotes. But they are called as a blue-green algae. That was an old name of that group. So now, where does a photosynthesis takes place? In higher plants, definitely that place is a chloroplast. Green areas where chloroplast is present, that is the site or place where photosynthesis take place. So take a chloroplast structure. Okay, so before moving into the structure of chloroplast, what about alignment of chloroplast within the mesophyll cells? And what about the number of chloroplasts? In the case of higher plants, particularly in flowering plants, if you go back to anatomy, under the heading leaf anatomy, in leaf mesophyll cells given, palisade parenchyma, spongy parenchyma. Within palisade parenchyma, a number of chloroplasts are present. Spongy parenchyma, less number of chloroplasts. So here, what about the alignment of these chloroplasts within the cell according to incident light? So alignment of chloroplast according to incident light within the mesophyll. The light which is available to the leaf. Available light, incident light. So for example, this is a mesophyll cell. Okay, so here intensity of light low light intensity. Low light intensity. Intensity of incident light is low. So that light is onto the surface of mesophyll cell. For example, this one is mesophyll cell. Mesophyll cell. So in this case, as the intensity of light is very less, what about the alignment of a chloroplast? Parallel to the wall. For example, this is a chloroplast. Parallel to wall. So if the intensity of light is incident light, available light is low, what about the alignment of chloroplast? Parallel to the wall. So chloroplast so in this case, maximum amount of sunlight is absorbed by the chloroplast. Reason? Intensity of light is very less. And against intensity of light, its arrangement is perpendicular. So here surface area is exposed to low light. So chloroplast is parallel to wall, cell wall, parallel to wall, and perpendicular to right angles, perpendicular to incident light. Wait. Here also take a parallel to incident light to harvest maximum sunlight. Next, second case, low light intensity, high light intensity. High light intensity mesophyll cell. So intensity of light is too much. If entire surface area is exposed, definitely that leads to damage in the chlorophyll. So in order to protect chlorophyll, more our intensity of light is more. Now it is perpendicular to wall and perpendicular to light also. Okay, 90 degrees. So chloroplast 90 degrees. Wall, concerned wall, and incident light also. Reason, intensity of light is too much. So that's why only some amount of some surface area should be exposed. If maximum surface area is exposed to that one, what about the chlorophyll subjected to denaturation at a high light intensity? So that is about arrangement of a chloroplast within mesophyll cells according to light intensity. Low light intensity, chloroplast parallel to that one. High light intensity, perpendicular to incident of light, incident light, and walls also. So that is about alignment of chloroplast. What is the purpose of this kind of alignment? Here, intensity of light is very less. To absorb maximum amount of sunlight, maximum surface area should be exposed to that. Here intensity of light is too much. Okay, so if maximum surface area is exposed to high light, incident light, what about chlorophyll? That will be denatured. So that's why only some part is exposed to that. So that's about alignment of chloroplast against incident light, available light. Okay. Next, what about the morphology of chloroplast? Structure of a chloroplast. So it is a double membrane bounded cell organelle. It is a type of a plastid. Here the given one is ellipsoidal or oval shaped chloroplast. In higher plants, the shape of the chloroplast is ellipsoidal, oval shaped. So this outer one, here envelope is with a outer membrane. This membrane is a lipoproteinaceous membrane, outer membrane and inner membrane. So these two collectively called as envelope. So double membrane bounded cell organelle, it is not part of endomembrane system. Okay, go back to cell biology, you'll get the information. Okay, so these membranes are lipoproteinaceous. Lipids and proteins are present. No pigments, completely without pigments. No pigment in the envelope. Okay, so envelope is with two membranes, chemically lipids, proteins, some extent polysaccharides, cholesterol also, but there is no pigment. So these are the membranes without pigments. Pigments are completely absent in outer and inner membrane. They are not concerned with harvesting sunlight and not concerned with converting so-called sunlight into ATP and NADPH plus. Okay, so reason that is because of lack of pigments. So what are the membranes without pigments? Outer, inner membrane of envelope. So here within this one, stroma part. Within the stroma, first one, a double-stranded circular DNA molecule. dsDNA, circular DNA molecule. Okay, so the genetic material of chloroplast is DNA molecule, double-stranded circular DNA molecule. Okay, so because of DNA, replication of chloroplast possible, 1 into 2, 2 into 4, doubling of chloroplast. So like how we studied a role of nucleic acid, that is autocatalysis, self-replication. Self-replication, and second one, self-replication means autocatalysis. Self-replication, autocatalysis. So autocatalysis is possible. Once autocatalysis is possible, definitely chloroplast is able to produce its copies. And second one, very, very important one, that is protein biosynthesis, heterocatalysis. Go back to molecular base of inorganics, heterocatalysis. What is heterocatalysis? Gene expression that includes transcription and translation, molecular basis of inorganics. Use that knowledge here. So as it is with a DNA molecule, transcription, translation possible, and various mRNA molecules are produced. Yes, autonomous cell organelle, I'll come back to that one. Because of DNA, it is autonomous, semi-autonomous. Semi-autonomous. DNA is able to produce a number of copies of it, means autocatalysis. So chloroplast is able to double if required. Transcription, translation possible, able to synthesize proteins and enzymes required for metabolic activities. And what is the type of DNA? Circular DNA. Circular DNA is generally present in prokaryotes. And what about a ribosome? 70S ribosomes. Ribosome is 70S. Only 70S are present. And if you observe carefully here and there, A chain of ribosomes are there. Here actually, somewhere you can take, no problem. So just a ribosome was given. That ribosome is a chain of ribosome. If you observe the textbook clearly, a chain of ribosome, that is polysome actually, polyribosome or polysome, attachment of a number of ribosomes to one mRNA. Go back to first year IP question. What is polysome? What about the importance of polysome? Okay, right? And here and there, starch granules are present. Lipid droplets are present. This one is a starch granule. And here and there, lipid droplets. So that is reserve food material. Lipid droplets. Okay, so this one is a stroma part. So within the stroma, what are the chemicals present? Inorganic and organic. Main one, genetic material DNA. 70S ribosome, membraneless cell organelle within the membranous cell organelle. So definitely they will ask you in chloroplast, transcription is within. What is transcription? Synthesis of RNA molecules from the DNA molecule within the stroma. So if they question you, autocatalysis, DNA replication, transcription, translation are within the stroma. 70S ribosomes. Okay. So as it is with a circular DNA particularly and 70S ribosomes, it is called as a prokaryotic cell within eukaryotic cell. As it is having DNA molecule and able to synthesize proteins and enzymes, other biomolecules required for metabolic activities, it is called as a semi-autonomous cell organelle. Wait. So within the stroma, from one end to other end, a number of lipoproteinaceous membranes are extended. Lipoproteinaceous, green colored lipoproteinaceous membranes. These membranes are well known to you. Like that, many are there. Okay. So this one actually that is green in color. So these are the thylakoids. So what about a thylakoid? Okay, so chemically, thylakoid is a lipoproteinaceous membranes, lipids and proteins and cholesterol and other biomolecules, majorly lipid and protein, lipids and proteins, lipoproteinaceous. Okay, with pigments. Pigments are present. We'll see what are the pigments later. So chemically, what is the difference between thylakoid and this membrane? Lipoproteinaceous, no difference. Without pigments, with pigments. And these envelope concerned membranes are not able to harvest, not able to convert solar energy into ATP and NADPH plus chemical energy, assimilatory power. But what about thylakoids? They are able to harvest sunlight and they are able to convert sunlight into ATP and NADPH plus. That is called as a assimilatory power, reducing power. So in thylakoids, thylakoid is a tubular membrane, tube, and that is a compressed tube-like structure. A tubular, for example, this is a tube-like structure. Compress it, press it. You will get a flattened tube, flat tube, simply. So thylakoids are flat tubes, flattened tubes. Tube means lumen is present. So they are flattened tubes. So tube means lumen is present. Lumen means empty space. Lumen means some room-like structure, lumen. So here thylakoids are of two types. One is Granum Thylakoid. Okay, so stacked thylakoids, a pile of coins or books, something, one above other. Granum Thylakoid. stacked thylakoids. So these are the Granum Thylakoids. And what about second one? Unstacked thylakoids are the stroma thylakoids, stroma lamellae. Okay, so stroma thylakoids are also called as intergranal thylakoids. So here, stroma thylakoids are able to connect, they are connecting simply. Grana 1, 2, in between these two stroma thylakoids. So within Granum thylakoids and stroma thylakoids, various pigments are present. Photosynthetic pigments are present. So that's why thylakoids are able to perform. So first one is light reaction. Light reaction. What is light reaction? It depends on sunlight. So during light reaction, sunlight, solar energy is converted into chemical energy. That is, what is that chemical energy? Chemical energy. This is radiant energy, sunlight, radiant energy, sunlight is converted into chemical energy. What is that chemical energy? ATP and NADPHH plus. So this is a assimilatory power, reducing power. With the help of ATP and NADPHH plus, plants are able to reduce C of CO2 into carbohydrate. Hence the name, reducing power. ATP and NADPHH plus. So light reaction is dependent on directly on light. What are the end products of main end products? Chemical products, ATP and NADPHH plus. With the help of so-called end products. Second one in photosynthetic mechanism is a dark reaction. Here, a dark reaction means that is not directly dependent on light. Indirectly depends on light phase. As it is indirectly dependent or not directly dependent on light phase, it is called as a dark reaction. So we should not construe that, understand that. We should not understand like this. Dark reaction is taking place in dark phase. That is a wrong statement. What is dark reaction? That takes place with the help of end products of light. So it is dependent on light indirectly, not directly. Okay. So dark reaction is dependent on light reaction indirectly. What is that indirectly? With the help of ATP and NADPHH plus. So dark reaction takes place within the stroma. So DNA is able to produce enzymes required for light reaction and dark reaction. So dark reaction is not independent of light, dependent on light. The question is whether it is directly or indirectly. That is the question. So it is indirectly dependent, not directly. Even without direct light, it is possible with the help of ATP and NADPHH plus. What is ATP and NADPHH plus? It is not independent, it is dependent. Indirect and direct, that is the question. Okay, so it is dependent on light indirectly, not directly. Okay, in the absence of light possible if ATP and NADPHH plus available. Okay, so that is about chloroplast. Definitely, they will link this chloroplast with molecular basis of inheritance. Under the heading molecular basis of inheritance, we studied transcription. If you go back to transcription, what about the number of kinds of RNA polymerase present in chloroplast? Only one kind. That codes for transcription of mRNA, ribosomal RNA, tRNA molecules. Okay. So that is about chloroplast. Now, what are the photosynthetic pigments? What are the pigments involved in photosynthesis? Photosynthetic pigments. Okay, so pigments are isolated, separated. Separation of pigments by paper chromatography. Go back to introduction part of plant physiology. Already we discussed what is paper chromatography. Okay, separation of pigments by one type of technique that is called as a chromatographic technique. So in chromatogram means chromatogram, a graph, a digital film, a graph that shows accumulation and presence of different kinds of pigments. Okay, chromatogram is a graph that shows various type of pigments present in that plant part. So according to chromatogram, what are the pigments present in higher plants? So first one, chlorophylls. Chlorophylls. So possible chlorophylls are A and B only in higher plants. In algae, A possible, B possible, C possible, D possible. And some are having E also, that is not required. As we are studying photosynthesis in higher plants, the question is what are the chlorophylls possible in higher plants? A and B only. Don't go for a chlorophyll D, C like that. Higher plants from bryophyta, pteridophyta, gymnosperms, angiosperms. They are with the chlorophyll A and B. And what about the color of chlorophyll A? Chlorophyll A is a bright or blue-green, bright or dark, simply blue-green in color. And chlorophyll B is a yellow-green, yellow-green. Next, second one, xanthophylls. Xanthophylls. And what about the color of xanthophylls? They impart what type of color? Yellow, generally, yellow color. And third one, carotenoids. Carotenoids. Okay, so carotenoids are yellow to yellow orange. Chlorophyll A, blue green in color. Chlorophyll B, yellow green. Xanthophylls, yellow in color. Carotenoids, yellow to yellow orange. Okay, so remember those are colors of concerned pigments. Okay, so out of all these pigments, the predominant pigment is a chlorophyll A. The universal pigment also chlorophyll A molecule. Okay, right? So that's about so-called pigments. And what is the role of these pigments? Okay, so first one, xanthophylls, carotenoids. First take these two things. Xanthophylls, carotenoids are generally called as these two. Generally, these two are collectively called as accessory pigments. Accessory pigments. Okay, so what is the role of accessory pigments? They are able to absorb sunlight. So whatever the sunlight absorbed, that is transferred to chlorophyll A molecule particularly. So what is the role of xanthophylls and carotenoids? One, absorption of light, sunlight, energy, and transfer of light to chlorophyll A, for example. Here I'm taking chlorophyll A. Okay, that is the main pigment. So they are able to transfer light to chlorophyll A. And the second one, protection to chlorophyll A molecule from photooxidation or photodenaturation. At high light intensity, chlorophyll A is subjected to damage, that is sensitive. So what is the role of accessory pigments? Transfer of light to chlorophyll A and providing protection to chlorophyll A from photooxidation, photo bleaching, or photodenaturation of chlorophyll A molecule. Actually, B also involved, but it was not given there. Okay, so these are the pigments involved in photosynthesis in higher plants. Next. Now, what about absorption spectrum and action spectrum of chlorophyll A, B, and carotenoids? So first one is absorption spectrum. What is spectrum? Spectrum of light. Absorption spectrum means light absorbed by pigment, simply. What is the absorption spectrum? So a graph that shows ability of pigments to absorb different wavelengths of light. A graph that shows, for example, this is a graph. Absorption spectrum. It's a graph that depicts or explains or shows the ability of various pigments to absorb sunlight at different wavelengths of light. Okay, so absorption spectrum of chlorophyll A, B, and carotenoids. Okay, so actually carotenoids are two types: carotenes and xanthophylls. Carotenes and xanthophylls. But according to textbook, chlorophylls, xanthophylls, carotenoids separately discussed. But here you have to take a chlorophyll A, B, and carotenoids. So what about the capacity of these pigments to absorb light at different wavelengths? What about the capacity of a pigment to absorb radiant energy? Okay, so take a 400 nanometers, 500 nanometers, 600 nanometers, and 700 nanometers of wavelengths of light. So here 400 to 500 are in between up to 516, 600. So the light wavelength between 400 to 550. So this zone is with blue light, and from here to here 600 above generally, that is red light. Okay, right. Now what about absorption capacity of first chlorophyll B molecule? Chlorophyll B molecule with a peak absorption. So this is a chlorophyll B molecule. Peak absorption. Two peak absorptions are there. One here and second one here. Okay, so highest absorption here. So chlorophyll B, this one is a chlorophyll B molecule with the highest absorption at blue light. And the second peak is at red light between 600 to 700. So how many peak absorptions are there? 1 and 2. 2 peaks are there. This one is the highest one and the second highest one. So peak absorption at blue light. And next highest one is at red light. So peak absorption 1, peak absorption 2, highest and least one out of these two things. So that's about chlorophyll B absorption. So between 400 to 500, what about absorption capacity of chlorophyll B molecule? More when compared with the same pigment at a red light or in between 600 to 700. Less here, but 2 peaks are there. Now, what about chlorophyll A molecule? So here chlorophyll A molecule with a peak absorption here. Wait. Chlorophyll A molecule with the peak absorption here, dropping down. Okay, so chlorophyll B also, here I'm taking, sorry, chlorophyll A, black colored one. So now take a 400 to 500, B with the highest absorption. And the second highest is a chlorophyll A molecule. So at a blue light, B with the highest absorption, second highest one, chlorophyll A molecule. Now, red light, highest A molecule, second one B molecule. Okay. Next, out of the comparison between A and A. At a blue light, A with the peak highest absorption. Next, 600 to 700 less. Overall, at a red light, 600 to 700, both are having less peak or not when compared with these two peaks. Okay, so now blue light, which one is with the highest absorption? B. Next, A molecule. Red light, A with the highest absorption, second highest B. Next, out of AA comparison, chlorophyll A molecule, more here, less here. Next, out of chlorophyll B here and B here, more here, less here. Okay, right? Next, carotenoids. What about the carotenoids? Generally, carotenoids are poor in absorption, generally. So what about the carotenoids? So peak absorption is at a blue light only. In red light, no absorption of light by those. So that is about absorption spectrum. What is absorption spectrum? A graph that shows the ability of light, ability of pigments to absorb different wavelengths of light. Okay, right? So that is about absorption spectrum. And second one, action spectrum. What is action spectrum? A graph that shows photosynthetic ability of pigments at a different wavelengths of light. More is the peak of absorption of light. Definitely. What about the rate of photosynthesis? More definitely. So here given one is a action spectrum of chlorophyll A molecule given. So what about a graph for that one? Okay, so here also 400 nanometers, 500, 600, 700 nanometers. And what about action spectrum? So more here, dropped and going back. Wait. Okay, so here that is about action spectrum. What is that action spectrum? If you observe that graph clearly, here given a statement, rate of photosynthesis, rate of photosynthesis with the help of O2 evolved. O2 evolved. Go back to Engelmann experiment. Engelmann experiment. If more amount of O2 is available, definitely more is the photosynthetic yield. For example, 6 O2 molecules, 1 glucose molecule, 12 O2 molecules, 2 glucose molecules. So this is a peak absorption, for example. Come back here. Peak absorption means more amount of oxygen is evolved. Definitely, what about the photosynthetic yield? For example only here 100 glucose molecules are produced. Glucose molecules. So highest rate of photosynthetic yield is at the blue light. And the second one, according to the chart given, 80 glucose molecules. For example only. Okay, second highest is in between 600 to 700. So highest, according to that one, highest is at blue light. There is no big difference between these two. Take slight difference only. For example, here 95 glucose molecules. Overall, little bit dropped or not. It depends on the amount of sunlight absorbed by concerned pigment. Here, A with more, A with less. Definitely more photosynthesis, less photosynthesis. Okay, so that's about action spectrum. And a third one, comparison between absorption and action spectrum of chlorophyll A molecule. Okay, so take one more graph and take a absorption spectrum of chlorophyll A molecule. This is absorption spectrum of chlorophyll A molecule. Chlorophyll A molecule, absorption spectrum. And what about action spectrum? Like this. Okay, so this red colored one is action spectrum of chlorophyll A molecule. Okay, so these two lines, these two curves, superimposed or not? Superimposed. Means one above another, not overlapped. If both are overlapped, they are not visible. If they are overlapped, for example, 4 fingers, only one is visible. If they are like this, 4 are clearly visible. This is superimposed condition, one above another. So in this case, come back. So here, this one is a absorption spectrum concerned curve, and this one is a action spectrum. These two are one above another, clearly visible or not, distinguishable or not. Are they overlapped? If they are overlapped, overlapping means, for example, this is a first curve, for example, and next one is like this. They are not clearly visible. Both are merged simply. So this is called as overlapping. Okay, so overlapping is wrong concept here. Take a superimposed condition. What is superimposed? One above another. Almost parallel or not? Both are parallel. Absorption spectrum and action spectrum of a chlorophyll A molecule. And definitely they will question you. What about the curves of absorption and action spectrum of chlorophyll A? Go for superimposed one above another without overlapping. Without overlapping. There is no overlapping. If overlapped, they are not clearly visible. It is superimposed condition of both the curves, right? And next one, what about the alignment of or organization of pigments within the thylakoid? How they are organized? Organization of pigments. So what about the organization of pigments? Pigment organization, how they are organized? Pigment organization. But actually the given heading in your textbook is what is light reaction. So before moving into what is light reaction, we have to know what about organization of pigments, how they are aligned, how they are arranged within the thylakoids. Okay, so pigments, various pigments, we'll see what are they. Pigments are organized into photosystems. Okay, so what about the photosystems? Photosystem 1, PS1 and PS2. Photosystem 1 and photosystem 2. Photosystem 1 and photosystem 2. So according to discovery, first discovered one, photosystem 1, and a second discovered one, photosystem 2. So the nomenclature of PS1, PS2 depends on the sequence of discovery or time of discovery. First one discovered, photosystem 1, second discovered one, identified one, photosystem 2, not on the basis of a sequence of involvement. In one electron transport system, PS2 is first involved, next involved is PS1. So the nomenclature of PS1 and PS2 is not on the basis of a sequence of involvement in light reaction. It was on the basis of a discovery only, first discovered and second discovered. So take a photosystem 1. So first one, PS1, photosystem 1. So in photosystem 1, two components are present. What are the components of a photosystem 1? So each photosystem with a LHC, first one, light harvesting complex. Simply LHC. Light harvesting complex. This is also called as a light harvesting system. Light harvesting system. Okay, and also called as, this is also called as antenna chlorophyll complex. Antenna, what is the role of antenna? Taking or observing waves or not, like that. So this LHC is with all pigments except chlorophyll A. So pigments, various types of pigments except chlorophyll A. Chlorophyll A is absent here according to that statement. All pigments except chlorophyll A plus pigments. So in LHC or antenna, pigments are bound to proteins. Sorry. Pigments are bound to proteins. Sorry. Pigments bound to proteins. So simply association of pigments and proteins in LHC, light harvesting complex. We'll see what about the role of that one after completion of components. And second one, that is light harvesting complex. This light harvesting complex is called as reaction center. That reaction center is always with chlorophyll A only. Okay, so these two are the two light harvesting complexes present in PS1. So in reaction center, for example, reaction center, simply I'm writing P, photosystem. Reaction center is 700. What is that 700? 700 means chlorophyll A molecule. What is 700? That is reaction center. This is a reaction center with a chlorophyll A molecule with a maximum peak absorption at a red light. So highest absorption, maximum absorption, saturation, high absorption of light at a 700 nanometers of red light. This 700 is nothing but a wavelength of light where it is able to absorb maximum amount of light or photons. What is photon? Energy particle. We'll see what is that photon. Okay, so that is about reaction center with chlorophyll, a molecule only, that is with a peak absorption at a red light with a longer wavelength. 700 is a longer wavelength, higher wavelength. So that is about a PS1. Next, what about a PS2? PS2, photosystem 2. Same as it is, simply change one thing, that is reaction center. Here reaction center is a 680. So peak absorption of a red light at 680. Here 680, remaining as it is. Okay, so each photosystem is with the LHC1, LHC2. Okay, light harvesting complex. This one is a reaction center. Now we have to check what is the role of both the complexes. Okay, so first complex. Now I'm going to erase the board. What is the role of a light harvesting complex is present in each photosystem? Okay, so this one is a reaction center. For example, this is the reaction center. Whatever the type of reaction center, this is a reaction center. Reaction center. The reaction center is a P700 or P680 chlorophyll A molecule. Here it is, the reaction center is with the chlorophyll A molecule. And it is surrounded by a number of pigment molecules bound to proteins. Okay, so this one is antenna. Antenna chlorophyll complex. Antenna chlorophyll complex is generally with carotenoids. Carotenoids plus chlorophyll B also. So what is the role of antenna complex? Okay, right? So sunlight. Okay, so photon energy particle. Photon energy particle. Photon is energy particle. It's some amount of energy. So initially, photon is absorbed by LHC. From there, it is transferred to reaction center. Okay, so now excitation of a reaction center and donation of electrons. So electrons are excited from the atomic nucleus of a chlorophyll A molecule here, and that electron is accepted by primary electron acceptor. So simply here we are discussing what is the role of LHC and a reaction center. Absorption of radiant energy by the LHC or antenna chlorophyll complex. From there it is transferred to reaction center. Excitation of electron, that is accepted by primary electron acceptor. Okay, so this is part of a mechanism of light reaction. We will study the mechanism of light reaction with the help of a so-called photosystem 1 and photosystem 2 at the time of light reaction, that is with electron transport system, cyclic and non-cyclic electron transport system. We'll see that one there. Okay.